Um, so we'll get started on our STEM World's Computer Science panel. So our first amazing guest is Dr. Anshul Kundaje, who is a professor of genetics and computer science at Stanford University. Dr. Kundaje completed his PhD in computer science from Columbia University. He has led analysis efforts for two of the largest functional genomics consortia, which are the Encyclopedia of DNA Elements and the Roadmap Epigenomics Project. The Kundaje lab develops statistical and machine learning methods to analyze large-scale genomic data. I'm very excited to hear about his research and use of machine learning and AI to analyze genomes. Without further ado, please welcome Dr. Kundaje.
Hello. Can you confirm that you can hear me?
Yes, we can hear you.
Excellent. Okay. So thanks very much for the invitation. I hope everyone's having a good weekend. So my lab's at Stanford, and we are part of the genetics and computer science departments. I'm going to talk to you today about how we use machine learning methods, particularly deep learning, to try to understand the control system of the human genome. So my lab works on the interface of biology and computer science, and it's a very exciting and interesting area. And I'll show you how we can use the combination of these technologies to understand basic biology, as well as to understand the genetic basis of diseases. All right, so jumping right in, I want to start off with what the human genome is. The genome is a long DNA biopolymer. It's made up of four units: adenine, cytosine, guanine, and thymine, ACGT. Believe it or not, it was only in 2003 that we, as humanity, actually obtained the first draft reference sequence of the human genome. By that, I mean the entire sequence of the genome, how long it is, what all the letters are. And we realized that the genome is actually, the human genome is about 3 billion nucleotides long. What's very interesting is, this was, of course, the Human Genome Project that almost spent over a decade trying to figure out the draft sequence. And it effectively cost about $3 billion. So the effective cost of sequencing one reference human genome, so this wasn't the genome of one particular individual, it was sort of a representative genome for all of mankind or humanity. That costs $3 billion for 3 billion nucleotides, right? So that's about $1 per nucleotide. Now, if you fast forward to today, there's been an absolute revolution in high throughput DNA sequencing technology. And in 2003, we started off with one sort of average reference genome for all of humanity. And now we can sequence individual genomes for exponentially lower costs. So today, you can sequence a genome in about a week, and it costs about less than $1,000. Also, the sequencing machines. So this shows like the super exponential growth in the number of genomes being sequenced since 2003. We are expecting by 2025 to have pretty much sequenced. Already, several countries have sequenced their entire populations. I expect that anyone who wants to get their genome sequenced voluntarily would be able to do it. It would be a routine part of, similar to how you get a birth certificate, you would potentially get a human genome by the year 2025. What's also been really interesting is the devices used for genome sequencing, which are often called high throughput sequencers. They start off being these giant, like, apparatuses that filled entire like football fields, and now they reduced over the years. This is 2008, this is 2009, this is 2012. These are like sewing machine models. They're pretty tiny, they can fit on your desk. And the latest is this Oxford nanopore sequencer. It fits in your hand, and if you look at it, it kind of looks like a USB device, right? You see this thing right there? That's because it is a USB device. It actually just plugs into your computer. You can swipe some specimen and start streaming DNA sequences right into your laptop. So this has really revolutionized what you can do with sequencing technology in the wild. So imagine there's an outbreak. As you know, we're living through a pandemic. There have been previous epidemics and pandemics in very remote regions. For example, the Ebola epidemic that happened. Imagine having to go and set up an entire lab there to just sequence, viral genomes or human genomes. It's much easier now because you're able to do it with these pretty small devices, which are very high throughput. So there's been an absolute revolution in terms of the cost, in terms of the throughput, and in terms of the size of the technologies. And I'm going to run you through how one of the most popular technologies works. This is called sequencing by synthesis. And the way it basically works is, DNA is a double-stranded polymer, right? So it has two strands, and they are complementary, which means the A's pair with the T's and the C's pair with the G's. So if you take one of the strands and you strip the other strand off, right, and you have a bunch of free-floating nucleotides, these A, C's, G's, and T's, let's say the different colors. If I take half, this stripped down single-stranded template of DNA, and I take a bunch of free-floating nucleotides and I add an enzyme called DNA polymerase, this enzyme will actually complete the complementary strand, okay, one nucleotide at a time. And what's really interesting is if I'm able to add different fluorophores to these nucleotides, so let's say my C's emitted a blue color, and my G's emitted a yellow, and my T's emitted a red, and my A's emitted a green, once they complemented with another strand, you can imagine that as the DNA polymerase is actually completing the synthesis of the strand, you will start seeing colored light, right? You will see blue, and then blue, and then blue, and then red, and then yellow. And so if you follow the sequence of colors as DNA polymerase is sequencing this single-stranded template, you could potentially read out the sequence, right? And that's the trick that sequencing by synthesis uses, okay? Now, of course, you don't want to do this one molecule at a time. You want to do this in an extremely high throughput fashion. And so the basic trick is you take the DNA from a person's genome, an organism's genome from the cell. You fragment it into millions and millions of little pieces. This is referred to as shotgunning the genome. And then you take these little fragments and you immobilize them on a template. On a surface. Okay, so each of these form templates. So you have millions of these templates on the surface. And then what you do is you add DNA polymerase with these colored nucleotides. And the main difference is that the DNA polymerase is synchronously completing the templates across the entire slide. Okay, so millions of different templates are all being simultaneously completed in a synchronous fashion. So in the first cycle of sequencing, the polymerase will add an A at this template and a G at this template, and you have a microscope sitting on top that's visualizing the template. And what it basically sees is something like this. So imagine you just had two templates. You typically have millions, but just imagine you have two. At each cycle of sequencing, if you look at those two pixels through the microscope, you would see, through the camera, sorry, you would see different colors, right? So in cycle one, you see yellow and green, then you see red, and then blue and yellow, and so forth. And so then you get a sequence of these colors at each spot, at each template. And once the sequencing is done, let's say there are 100 cycles, you get a set of 100 colors for every spot, a sequence of 100 colors, and that basically gives you the sequence, okay? So that's the trick behind high throughput sequencing. You take these billions of DNA molecules, you immobilize them on a surface, you synchronously sequence them with fluorophore nucleotides, each of them having different colors, and a camera reads off each pixel at a time, basically decoding and telling you what the sequence is. So this is really cool. It's really fast and really high throughput and very accurate. And what you can do is start using this to basically just sequence individual genomes. So now no more, let's not talk about the reference human genome for all of mankind or humanity. Let's talk about individual specific genomes, right? So your genome and my genome. And why this matters is that all of us have two copies of each chromosome. So we have 22 pairs of autosomes and then two sex chromosomes typically. And so what happens is each of us has two copies of the genome, as shown right here. And all of us differ at specific positions. So for example, this individual here has a T at this position and an A at this position in the two copies. Okay. Another individual might have an A and an A. Another individual might have a T and a T at this position. Okay. And so these are called genetic variants. These are positions at which our genomes actually differ. This is what actually makes us different from each other. Identical twins would have exactly the same genomes. That's the only case where two individuals have exactly the same two copies of each nucleotide. But what we can do is now start sequencing individuals and sequencing their diploid genomes, these pairs of genomes, and trying to identify all the positions in the genome where we see genetic variation between individuals. And most of this variation is not harmful. It is, in fact, very good for us. It creates the diversity that we see. And by sequencing a healthy individuals, we can start doing really interesting things like ancestry inference, tracing back our history, uh, very interesting things just to study basic biology, genetic variation, and so forth. Now, another interesting application for disease is you can use the same technology to sequence not just healthy individuals. So here we are showing a collection of healthy individuals, often referred to as controls, but you can also sequence a bunch of individuals who have a specific disease. Okay, and these are called cases. And so now what you'll see is that specific positions in the genome, the controls have particular patterns of nucleotides, and the cases have other patterns of nucleotides. And by comparing for at each position in the genome, you can do a statistical test. Basically, you just enumerate how many individuals have one particular pattern, how many individuals have the other pattern, and the third pattern, right? And if the healthy individuals have a very specific enrichment of one pattern with respect to the diseased individuals, then that particular genomic locus, that variant, has likely something to do with the disease. It's basically genetically associated with the disease, right? And so this is a very powerful way of mapping genetic variants that have some association with the disease, which may cause predispositions to disease. Okay. And here's one example for Alzheimer's disease, which, as you know, is a neurodegenerative disorder. This is referred to as a genome-wide association study. It's basically this, but you perform this kind of test, this kind of table for every genetic variant in the genome. Okay, and so far we've found millions of genetic variants in the genome by sequencing human populations. And so here what you're seeing is the output analysis of a genome-wide association study for Alzheimer's disease. On the x-axis is the whole genome, okay, 3 billion positions. On the y-axis is the statistical significance of association. This just tells you basically how strongly associated each genetic variant is with the disease, in this case, Alzheimer's. Each dot on this plot is a genetic variant. And so you see most of the genetic variants have very low association, right? So they're not having anything to do with the disease, but then a bunch of them really skyrocket right up here. And this kind of plot is referred to as a Manhattan plot because, as you can see, it kind of looks like the skyline of Manhattan, right? You have these skyscrapers popping out of thin air. And these are the genetic variants that are likely predisposing individuals to Alzheimer's disease, okay? So it's a very powerful way to map. You can take any disease of interest, any trait. It doesn't have to be a disease. It could be like hair color, eye color. You can do the same kind of studies. You sequence a bunch of individuals which have one property of the trait versus another, or one disease status versus the other, and you perform these association tests, and you can map these genetic variants associated with the disease. So this is really powerful because now we've been able to map thousands of genetic variants that associate with all kinds of diseases, complex diseases, type 2 diabetes, cancers, Alzheimer's, you name it. The issue is once we've identified these variants or these loci that do interesting things, we don't know what they actually end up doing. Like, okay, we found an A and a C that associate with Alzheimer's, but how is it predisposing you, right? So that's where we come in. My lab really focuses on decoding genome function. And by that, what we mean is if you take a particular letter in the genome or a particular word in the genome, our goal is to try to figure out what it actually does. Like what is its function? What is it encoding? Okay, that's called functional genomics. And it's very complicated because when you're interpreting the genome, you remember that the human body humans are multicellular organisms. So we have a whole host of um, tissues and cell types within those tissues in the body, tens of thousands of different cell types. And what's amazing is for me, for example, if you take any single individual, all the cells in that individual's body have the exact same genome sequence. Okay, even though they have entirely different functions and morphology, they have the exact same sequence. So the big question is, how does the same sequence manifest into all this beautiful diversity of cell types and function? And so when you're interpreting the genome, you can't just interpret it in a vacuum. You have to interpret the genome in the context of which cell type or tissue you're interested in. So for example, if you're thinking about Alzheimer's disease, it is a disease of the brain, and that means how to interpret the genome in the context of brain cell types, okay? So this complicates the whole picture. So to understand the function of the genome, we first have to start dissecting genome biology a little bit. Okay, so let's take the cell, let's get into the nucleus, let's unwind some DNA, and let's start looking at what are the different kinds of functional elements in the genome. So many of you have heard of, I'm sure you've heard of genes. The human genome has about 20,000 genes that encode for proteins. So you can read off the sequence of the gene, and it literally is a code for a protein. It produces you read off the DNA sequence, and it exactly translates into a corresponding sequence of amino acids that form a protein. So there are 20 to 25,000 protein coding genes in the genome. But the protein coding sections of the genes only account for 1.5% of the 3 billion nucleotides. Okay, so all the proteins in the genome, I mean the cell, are produced from 25,000 genes that occupy the protein coding parts of it, occupy only 1.5% of the 3 billion nucleotides. So what does the rest of the genome do? And how are these genes actually controlled? Uh, the secret to having different cell types and tissues and function is to express different sets of genes in different cell types. So you can have 25,000 things, but you can combine them in many, many, many different configurations, thereby giving rise to very, very distinct cell types and tissues. But now if I want to activate gene A only in brain and not activate it in liver, there needs to be some control system, right? Telling the cell, how is this gene active in cell type A, or activate it now, don't activate it now. And so where is this control system? What's really cool is the control system is also in fact encoded in the genome. There are a bunch of genomic elements called regulatory control elements. These are little sequences of DNA, and they are the on-off switches. They tell the genes to turn on and off, or they contain sequences that tell genes to turn on and off. And if you look at the anatomy of any of one of these control elements, what actually happens is they are bound by a variety of different proteins. So a bunch of these proteins that are produced by genes actually have regulatory function. So they go back and bind these control elements. And when they bind these elements, they basically activate genes or repress genes. Okay. So the whole language of these proteins, these regulatory proteins that bind DNA, and a whole other collection of biochemical flags. These are like methylations and acetylations, all kinds of biochemical flags that can be added and removed on the DNA. And that makes the DNA dynamic. So even though the DNA sequence is static across all the cells in the body and across the entire lifespan of an organism, the way the genome is expressed and controlled is dynamic. And the way that happens is through the dynamic binding or unbinding of collections of proteins to these regulatory elements, and the addition and removal of these various kinds of biochemical marks. And what's really cool is we can use high throughput sequencing technology that I showed you before to profile all of these different biochemical events across the entire genome. So we can perform experiments that give us these beautiful digital readouts across the entire genome. At each nucleotide in the genome, we get a readout of whether a particular protein is bound. For example, this black protein right here, is there a biochemical modification or not? And so we see this really beautiful painting of the genome sequence. And this painting, this sort of multi-dimensional painting of the genome by all of these different proteins and biochemical marks is cell type specific. Each cell type has a different painting of the genome, okay? And that really represents its dynamic instantiation. So if I have all of these different paintings of the genome in different cell types and tissues, I need to map these. And so what the National Institute of Health did is they, just as they funded this massive human genome project, which was a beautiful, large collaborative effort across many different countries and universities and industry and academia, same way the NIH funded two other large consortia, one of which is called ENCODE, the other is called the Roadmap Epigenomics Project. These started off immediately after the Human Genome Project and recently have completed major portions of their goals. And what they've ended up doing is these projects are profiling these hundreds of different molecular markers, these paintings, as I showed you, right? Different proteins binding DNA, different biochemical modifications, hundreds and thousands of these across the entire genome in hundreds of different cell types and tissues, okay? So you end up with this amazing teracube of information, digital readouts, which tell us which molecular flags are present at which position in the genome in which cell type. And this is a terascale cube of just numbers. And so we need to figure out what it means. And that's where we come in. And that's where computer science comes in and machine learning. And these data sets are massive. And just as Google and Facebook use machine learning to study, the massive amounts of data on the internet, similarly, we can use machine learning to start understanding patterns in the genome by analyzing these kinds of teracubes of information. Okay. So what did we learn from this? The first thing we learned is we are able to now paint the genome. So this is one small portion of the genome, okay? And this is a gene called PAX5. You may ask, what does PAX5 do? One way to answer this question is to perform these experiments in hundreds of different cell types and tissues and paint the genome. So we literally color the genome, each position in the genome with a different color, and the colors correspond to these different markers of activation, repression, proteins, binding DNA, different biochemical markers. And what we're showing you here is a compressed representation of all these hundreds of different markers. So wherever you see red, it's an active control, red or yellow, it's an active control element. If you see green, it's the body of an actively expressed gene. And if you see gray, it is just a repressed region of the genome. Okay. And now we can do this in every cell type or tissue in the body. So what I'm showing you here is 5 or 6 cell types. These are embryonic stem cells, different types of embryonic stem cells. And you can see that this gene PAX5 is gray in color, right? So it's repressed. It's not really being activated. Now, as I move to different cell types, so there's the same piece of the genome, and I'm seeing how it's being painted now in blood cells, okay? T cells and B cells. And then I can do this in hundreds of other cells. Epithelial cells, brain cells, muscle, heart, smooth muscle, digestive, cancer cell lines, all kinds of stuff. And notice what happens to this gene in very specific cell types. These are specific types of blood cells, immune cells. You see the start of this gene, the control element turns on, it goes from gray to red, and the gene body fires, right? It goes from gray to green. And so this way we can really not just get genome sequences, but we can start figuring out dynamics of gene activity and control element activation, all these beautiful yellow things that are popping on and off in different cell types and tissues. And so we get this beautiful dynamic map of genome activity in hundreds and thousands of different cell types and tissues. And what we realize is for the first time, we're able to map the control circuitry of the genome, and just 25,000 genes are being controlled by more than 2.5 million control elements, okay? And that really gives rise to the complexity of the human phenotype. It's not that we have many, many more genes. We have just 25,000 genes, but we can control precisely how those genes are expressed in different cell types and tissues. And there are millions of control elements turning on and off in different cell types and tissues, controlling the activity of these genes. So this is great because now we're able to map the precise elements in the genome that are controlling gene expression for the first time comprehensively in hundreds of cell types and tissues. But the big question is, how is the DNA sequence of these elements encoding this information? And so that's where machine learning comes in, okay? And the key thing to remember again is if you take any of these control elements, they're bound by these proteins, right? you must be wondering, how do the proteins figure out they're supposed to bind these locations, right? The genome is huge and vast, and why does this protein, green protein, come and bind this specific spot in the genome? It's because these proteins recognize very specific sequences of letters. We'll refer to these as DNA words or motifs. And what's amazing is that these control elements have their own language. Just like we have languages with words and syntax and grammar, the genome has its own language. It has a whole bunch of these beautiful DNA words that are found in these control elements in very specific syntax and grammars. And that really drives specific combinations of proteins to come and bind those words, thereby activating genes. Okay? So our goal is to decipher the language of the genome, the words and the syntax and the grammar. So how do we do that? Well, one way to do that is to basically convert this whole problem into a machine learning prediction task. Okay. And the basic idea is, given one such high throughput sequencing experiment, right, where I get these readouts of, let's say, this protein binding DNA across the whole genome, one such experiment gives me a beautiful readout of the likelihood that the protein is bound in those locations. Okay. So what I can do is I can take the whole genome, which is 3 billion letters, I can chunk it into little bins, let's say of size 200 base pairs, 200 letters. And each of those base pairs, right, each of those sequences, sorry, get assigned a value, a label. And the label tells me, is that region bound by the protein or not? Or it tells me some real valued number telling me how strongly the protein is binding that region. And so what I end up with is a machine learning problem called a classification task. Think of this as similar to like how when you search for images on Google, they are able to very quickly like label images like cats, dogs, elephants, whatever it is. It's the same kind of problem here. Instead of mapping images to labels, as Google does, we're trying to map sequences to labels. Our labels are not cats and dogs. Our labels are, is a protein binding the sequence or not? And so we have millions of these sequences across the genome, right? Every chunk of the genome is a little sequence, and the experiment assigns it a label telling you, is that sequence bound by the protein or not? And this is a beautiful machine learning problem called a classification task. Our goal is to take a bunch of sequences Si and to map them to corresponding labels Yi through a function, a mathematical function that takes these sequences and somehow transforms them into a label. Okay? And this is called a supervised classification task. That's where neural networks come in. So neural networks are basically machine learning algorithms. They are very powerful pattern detectors. What they can do is take one type of input, like an image, and map it to some other type of output, like a label. In this case, our inputs are sequences of A, C's, G's, and T's. The neural network has a whole bunch of these neurons. Neurons are just pattern detectors, and there are layers of these neurons. And these layers of these neurons learn increasingly more complex patterns as you go up the layers. The neural networks are kind of based on the, loosely, not very strongly, but loosely based on the structure of the brain and neurons in the brain, neural circuitry. Neurons are basically pattern detectors. And the neurons that are closest to the sequence right here, they learn very simple sequence patterns. And the neurons in the next layers take the patterns from the previous layers and learn more and more complex patterns. Okay. And the last layer of the neural network tries to predict these profiles from the experiments. Okay. Either a binary label telling you that the protein is bound or not, or it tries to predict the actual signal. Another good analogy to think about this is like a text to speech converter. Remember, like, so you have a lot of these software that can take text and generate speech out of it. That's also usually a neural network. In this case, our text is the DNA sequence, and our speech is the signal that's coming out of the genomic sequencing experiment. That's telling us whether a protein or some biochemical marker is present at a particular position or not. And these models are incredibly accurate. So firstly, they learn patterns from the sequences de novo, right? And secondly, they can very, very accurately predict these patterns. So here I'm just showing you some examples. These are observed, these are the experimental patterns, right here, this red one, and this one is a predicted pattern from the neural network. You can almost not make out the difference. The neural network is just able to take the sequence of that region in the genome and predict this beautiful profile coming out of an experiment. Now, this gives us huge power because now that we have this model that has taken sequence and can learn to predict what these profiles are, we can start interpreting these models. What are the patterns the models are learning? So what we do is we take the model that is trained, and then we interpret which nucleotides in the input sequence are actually giving rise to the predictions. And what we can do is take any genomic sequence. Here's a regulatory element. It has thousands of nucleotides, and every nucleotide in that sequence gets a score. And the score tells us how important that nucleotide is to predict this output. And you can see that in this particular regulatory element, most of the nucleotides are not doing anything particularly important, but this one is. And we've identified a DNA word, this TGCAT thing. This is the binding site of a protein called OCT4. Okay, so this is amazing. The neural network can just de novo take raw sequences, map them to these experimental profiles, and using computational methods that we've developed, we can interpret the models and identify the DNA words and their syntax. That means who comes next to who, what's the spacing differences, what should be the arrangements, and so forth. And we can do also very powerful in silico experiments, mutation experiments, where you can mutate these sequences and try to understand how important they are for biological function. So I'll show you one example of how we actually validate these predictions. I'm sure many of you have heard of CRISPR experiments, CRISPR-Cas9. So CRISPR experiments are these new genome editing technologies. These sequencing readouts that I showed you before, think of these as ways to read the genome, right? Because we're able to sequence the genome, we're able to read all of these biochemical markers, but can you also write the genome? Can you actually edit the genome? CRISPR is one of these technologies that allows us to go into cells and actually edit DNA sequences in actual cells. Of course, we don't do this in humans. That's a no-no. We do this in like cell lines in the dish, or in cells in a dish. But here's an example of what how we can use the computational models to predict what a mutation would actually do to these biochemical profiles. Okay, so here's a predicted profile from the model for one particular DNA element. Here's the DNA word that the model predicts as being important. Now, if we perform the experiment, this is what it gives us. So this is coming from an actual experiment. This is what the model predicted. Okay. And you can see they're quite remarkably alike, right? I mean, there are slight differences, but they're really similar in terms of shape. Now, what we can do is in the computer, we can mutate the sequence. We can convert these TTs to AG. And now the model predicts this red profile. Notice what happens. The red profile is much smaller at this position. And there's also attenuation of the profile at this position. Now, this is computational, okay? There's no experiment. The neural network is predicting what would happen if you had a mutation at this position. Now we can actually go into the cells and mutate these two nucleotides and then measure the readout. And this is what we get, okay? You can see it again is beautifully corresponding to what we see the model predicts. So this is amazing because now we have a model that you can make millions of mutations in the genome sequence, and the model can computationally predict what would happen to all of these molecular readouts. And so using this strategy, we can now take all the 3 millions of genetic variants that we found in the genome, and in each cell type, we can predict the effects on different kinds of molecular readouts, thereby giving us a way to figure out function of these variants. So in the last 1 minute, I'm just going to really quickly show you how we do this for Alzheimer's disease. I'm going to skip this slide. Again, this is the Alzheimer's genome-wide association study. You have all of these loci, right? These genetic variants that show really strong predisposition to Alzheimer's. What we can do is take the brain and perform these kinds of experiments that I just showed you, these molecular profiling experiments, in all the individual cell types in the brain. The brain is a complex tissue made up of many, many different cell types. And here's one such example of a genomic region. We are seeing different types of cell types in the brain: neurons, microglia, which are the immune cells of the brain, astrocytes, oligodendrocytes. There are hundreds of different cell types in the brain. And you can see the same region of the genome can have different activity in these different cell types, right? So you can see, for example, this region is very specifically on only in microglia, right? You can see a strong signal in microglia, but it's off in all the other cell types. We can fit our neural network models, and then we can take those genetic variants that are predisposing individuals to Alzheimer's, and we can use them you can push them through the model to see what they actually do. And here's an interesting prediction. Here's one of these genetic variants. These typically have these kind of weird names, RS some number. In microglia, in these cells, microglia cells, what our neural network predicts is that this genetic variant sits within a DNA word, this particular DNA word, GGG, CGAGG. It is the binding site of a protein called KLF4, very important protein. And when this G is mutated to an A, which happens in the diseased individuals, notice what happens. The model predicts the DNA word is decimated, and the binding of this protein is decimated, and the nearby genes get dysregulated. Okay. And so we are now able to interpret genetic variation. We're able to tell that this variant is important for Alzheimer's, but we know what it's doing to some extent. At least we can predict what it's doing. It's disrupting this DNA word, which is a binding site for this protein. And it's the A that specifically does the disruption, right? So we even know which letter, which mutation actually creates the disruption. So we can do this for all the millions of variants in the genome and start interpreting what all of them do, how they break DNA words, which proteins bind to them that are dysregulated, which genes nearby are dysregulated, and in which cell types the dysregulation is happening. So here's the cool thing. Even though Alzheimer's is a neurodegenerative disorder where our neurons basically end up dying, this genetic variant is not actually affecting neuron function. It is affecting microglia function. Microglia are the immune cells of the brain, which interact with the neurons to keep the brain healthy. Okay, so this gives us very, very specific predictions to go after therapeutically. Okay, and so just to summarize, we can use large-scale molecular profiling experiments with DNA sequencing technology to decipher genome function. Machine learning models or molecular profiling data can be used to interpret and decipher functional DNA words and syntax. The language of the genome can be validated using genome editing experiments. Molecular profiling data and machine learning models can be used to decipher mechanisms of disease-associated mutations. And hopefully I've convinced you that computational biology is a highly interdisciplinary science that is awesome. You get to do computation, you get to use really amazing concepts from computer science and machine learning, and at the same time jump back to the wet lab, go back to actually doing beautiful experiments with real life cells, use some of the greatest technology that exists today with high throughput sequencing, and really make inroads to understand disease mechanisms. And with that, I'll just end by saying that all the hard work that I just showed you today has been largely done by my students and postdocs in my lab. So all credit goes to them. And none of this happens in a vacuum. The government, particularly NIH, funds a lot of this work. And so I thank them for their contributions, and also our countless collaborators. Science is no more something that can be done by any single individual, any single lab. It is a collaborative effort, and it's a ton of fun to work with a whole bunch of really smart individuals. And with that, I will end. Thank you.
Thank you so much, Dr. Kundaje. That was awesome. We don't have time for questions right now because we're a little bit behind. We're very behind schedule, just with all of the events since, we've had events since 8 a.m. this morning. So if you're watching the live stream, just know we're a little bit behind.
Yeah, no problem. What people can do is either just type on the chat if you have a question. I'll stick around for a bit, or you can, I'm on the Slack as well, so I can answer questions there.
Okay. Thank you so much. Thank you so much, Dr. Kundaje.